Now, for real this time, we are recording. Okay, so on the first day of class, I told you that AMP2 is kind of a step up from AMP1. It's more than just, learning about the content. It's more about actually fitting these systems together to form this kind of mental model, so that you can easily diagnose patients, so that you can easily recognize the symptoms of patients and move forward with the diagnostic process or with the treatment process. Part of that, of course, is these articles that will be starting over the weekend and into Monday. The other part is case studies. And so I'm going to give you some information about a patient now, and then we're going to go forward with our lesson, and then we'll come back to Jennifer and kind of talk about what is going on with her after we know a little bit more about red blood cells. Okay, so Jennifer is a 37-year-old Caucasian woman who presents to her gynecologist complaining of, there we go, 2 months of intermenstrual bleeding, so bleeding between menstrual periods, and heavy, prolonged menstrual flow. She is really tired and very weak since her last pregnancy, which ended about 4 months ago. So we can see that she has a small child here. And so this would be in addition to the normal fatigue that a postpartum woman would experience. She has shortness of breath with exertion and craves cold celery for the past 3 weeks. And so this might seem to be really out of place here, but in fact, all of these tiny little details, which might not immediately come up in an interview with a doctor, might come up if you are an occupational therapist or if you are any kind of individual nurse, MA, working with the patient, right? So craving cold celery for the past 3 weeks. Also, some other details here. She has long-term NSAID use. So these are non-steroidal anti-inflammatory drugs like aspirin or ibuprofen that she's been using for knee pain. Okay. Of course, her gynecologist sends her to get blood tests. And so these are some of the results. Remember, last week, we talked about hematocrit, HCT, so the percentage of red blood cells out of whole blood, RBC, so red blood cells, hemoglobin, white blood cells, and then some other solutes within the blood. Okay, so at this point, we should start to become familiar with the appropriate levels for these different factors in the blood. Okay. Also, this is a blood smear. Okay, so just like we looked at last week in lab, this is the reference, so this should kind of look familiar. We see lots of nice bold red blood cells, and of course this here is a neutrophil, and this is Jennifer's blood smear. Okay, so just keep that in mind. We can see some differences right off the bat. Okay, so let's learn a little bit more about the red blood cells. Last class, we ended by talking about hematopoiesis, right? So the making of all of the formed elements of the blood. We talked about thrombopoiesis, making platelets. We talked about leukopoiesis, making white blood cells, which of course we'll talk about in a lot more detail in the immune system. And now we get to erythropoiesis. So this is the creation of erythrocytes, or the red blood cells. And so erythropoiesis is kind of happening constantly within our body, but it increases in response to what is called hypoxia. And note that hypoxia is blue and underlined, so this is a term that you guys should be familiar with for upcoming assessments. And so hypoxia means insufficient oxygen, and this can be caused by all sorts of different things, right? So when you first start exercising, you don't have quite enough oxygen in your blood. When you move to high altitude, or you go on vacation in high altitude, all of a sudden there's less oxygen in the air, and so you don't have as much oxygen in your blood. Or this can be because you are losing blood, and therefore you have less oxygen than you should have within your blood because it is leaving your body. And so hypoxia, for whatever the purpose of it is detected in the kidneys, and to a lesser extent the liver, but primarily this process is controlled by the kidneys. The kidneys, as we'll learn, are a powerful endocrine organ in and of themselves, and when the kidneys filter the blood and they detect that there's not quite enough oxygen, they are going to release a hormone called EPO, or erythropoietin. And so this is the hormone that, of course, is going to circulate within the blood. And ultimately, as that blood arrives at the bones, right, in the bone marrow, it is going to bind to receptors on those stem cells, and it's going to stimulate those stem cells, right, the hemocytoblasts, to proliferate, so to undergo mitosis, so making more and more and more of these cells, and to ultimately for those daughter cells to start differentiating. So that is to start expressing red blood cell genes as opposed to megakaryocyte genes, as opposed to white blood cell genes. Okay, and so within the next day or two, the red blood cell content, or kind of the precursors to red blood cells, are going to start increasing in the blood, right? So it only takes about a day or two after EPO is released to a certain extent to actually increase the content of red blood cells. Okay, and so this diagram here gives you actually quite a few different steps in the process of erythropoiesis. I am going to focus on just a few of them. You don't have to know every single one of these terms. Okay, so first of all, this entire process is happening within the bone marrow. Remember that that location shifts a little bit over the course of your life, but in general, as an adult, your bone marrow is where this happens. All red blood cells, erythrocytes, are ultimately derived from multipotent hematopoietic stem cells, right? So these guys can become any of the formed elements. Okay, given EPO, the daughter cells of these hematopoietic stem cells are going to differentiate into myeloid progenitor cells. From there, EPO is going to further commit these daughter cells to becoming a red blood cell. And again, this differentiation process only moves forward, never backward. Once certain genes are turned on and others are turned off, there's no reversing that. So these pro-erythroblasts are really the first cells that are absolutely going to grow up to become red blood cells when they get bigger, or smaller in this case. And so these are absolutely committed to being a red blood cell. Again, this is in response to EPO released by the kidneys, and this differentiation process is tied to turning on of some genes and turning off of some other genes. Now from here, these proerythroblasts these proerythroblasts require a few different ingredients in order to ultimately become a red blood cell. Okay, a couple things that they need are vitamin B12 and folic acid. Vitamin B12 is a vitamin that you acquire through your diet, specifically eating animal products. B12 is absorbed in your intestines, into your blood, and ultimately into this process. But in order to be absorbed, it requires something from the stomach to be secreted, and that something is called intrinsic factor. We'll talk about that as we go forward, but essentially we absolutely need vitamin B12 in order to synthesize the DNA needed to make red blood cells. Okay, so B12 is required for red blood cell creation. If you don't have enough B12, that is, if you suddenly become a vegan and don't start taking supplemental vitamin B12, over time your reserve of B12 is going to go down and you won't be able to make enough red blood cells. Okay. Also, as this cell is continuing to develop, right, it's using vitamin B12 to ultimately make the DNA for these cells. At about this stage, so the polychromatic erythroblast, at this stage, we are actually making abundant proteins, right? So over here, we're making the DNA, okay? By this phase right here, we start transcribing and then translating different genes within that DNA, which will ultimately make the protein called hemoglobin. Okay, so hemoglobin you've surely heard of before, but this is a protein which contains 4 different protein subunits. Right, and so if you look in here, you can see the alpha helices, and so this is just a chain of amino acids. all of these are grouped together to form a functional hemoglobin molecule. If there is any mutation in any one of these subunit genes or a problem with the translation process in any of these proteins, you get a defective hemoglobin. And so defective hemoglobins cannot carry oxygen appropriately. Hemoglobin also, in addition to these two pairs of protein subunits, it also contains 4 what are called heme groups. And so we can see hemo and globin making up the hemoglobin molecule. Okay. The hemes are actually iron molecules whose sole function is to bind to oxygen. Okay, and so there are 4 irons and 4 protein subunits in each and every hemoglobin molecule. And again, these hemoglobins are just being cranked out one after the other until the entire kind of immature red blood cell is filled with hemoglobin at this particular stage. Okay. In order to actually make hemoglobin, you not only need the DNA, you not only need the amino acids to build the proteins, but also you need to have enough iron. And so when we look at blood test results, oftentimes you will see iron levels and ferritin levels. Okay, iron itself, of course, is directly incorporated into hemoglobin. And so these are the appropriate levels, 50 to 175 micrograms per deciliter. That is the healthy range. We talked about transferrin last week. Transferrin essentially transports the iron throughout the blood from one place in the body to the other, so it can be recycled into hemoglobin. Ferritin is similar but different. Ferritin's job is to actually store the iron until it is ultimately needed. And so ferritin levels, of course, vary between males and females. But again, these ranges are healthy. If iron falls below these ranges, that might inhibit your ability to create red blood cells. Okay, to go back here just a moment, EPO has stimulated the differentiation of these myeloid progenitors to becoming pro-erythroblasts. Pro-erythroblasts are going to become red blood cells. They cannot become white blood cells. They can't become platelets. Okay, it's committed at this point. Certain genes are turned on, a lot of them involving hemoglobin. And so hemoglobin is created as long as we have enough vitamin B12, enough iron, and enough, of course, amino acids to make the proteins. Okay, so that is what's happening here in the polychromatic erythroblast phase. Okay, ultimately, these cells are going to be pretty much plasma membranes chock full of hemoglobin. Okay, once there is abundant hemoglobin created inside the cell, we can pretty much get rid of all of the other organelles, right? We don't need a nucleus, we don't need a Golgi apparatus, we don't need any more ribosomes. None of those things are going to become or going to be important in the life of the red blood cell. Okay, the red blood cell's job is to transport oxygen, nothing else. And so pretty much we want to get rid of all those other organelles so that they're not taking up space. They're not making the cell really big and clunky and difficult to actually make it through things like capillaries. Okay, so once we have enough hemoglobin, we actually kick out, right, enucleation, all of the other organelles, leaving behind this sac of hemoglobin only. Okay, at this phase, we call the immature red blood cells reticulocytes. And so that's the word way down here at the bottom. Reticulocytes are immature red blood cells. And these are actually the ones that are released into the blood circulation via those sinusoid capillaries in the bone marrow. Okay, note that this word is blue and underlined. It's one that you really need to know, right? You don't need to know pro-erythroblasts or polychromatic, none of those things, but reticulocyte is really important to know. Okay, again, once EPO is released, within a day or two, the reticulocyte count is going to increase substantially. Okay, we can also measure the reticulocyte abundance in your blood. And so usually of all the red blood cells, 1 to 2% of them are still immature. They're still reticulocytes. If you see much higher levels of reticulocytes, that means that you're producing red blood cells more quickly than expected. Okay. Any questions about this? Okay. Okay. So back to hypoxia. Why would this process be increased? For example, insufficient hemoglobin, right? So why would you have low oxygen? Well, if you don't have enough hemoglobin, you're not going to have enough oxygen. And so your body is going to start producing more blood to compensate for that lack of oxygen. And so if you have low iron, if you have low B12, right, you're not going to have enough hemoglobin. Okay. As I briefly mentioned, high altitude, right? So when you go to the mountains on the weekend, of course, from sea level by Stockton, of course, there is less oxygen in the air. And so when you first get there, it's going to be really hard to breathe, and you might get a headache, and you might get kind of dizzy when you stand up, right? All of that is because of hypoxia, because there's not enough oxygen in your blood. And so this will actually stimulate your bone marrow to start making more and more red blood cells so that you can breathe better within a few days, right? So that's why you don't climb Pikes Peak. The day you get to Colorado, you have to wait a little while for your body to catch up to the new conditions. And this is me in the corner on the north rim of the Grand Canyon a few years ago. That's about 9,000 feet in the 9,000 feet in the air. So pretty hard to breathe, especially after coming directly from South Jersey. All right, so what these up here are representing centrifuged whole blood. Okay, remember I told you that when you remove whole blood from the body, you treat it with an anticoagulant and you spin it really quickly, we get the separation of the plasma and the red blood cells, right, or the hematocrit. Remember that on average, 45% hematocrit is normal, right? And of course, this varies between males and females, but 45% is generally accepted, okay? When you have more red blood cells than 45%, right? Again, on average, so when you have high red blood cell count, this is called polycythemia. Again, big clunky word, but essentially poly, many, cyth, cells, emia, in the blood. So you have many blood cells in the blood. And so this can be indicative of disease, and we'll talk about that more in a bit. But this is actually a completely normal response to being at high altitude, right? So within a couple days of being in Arizona, if you were to look at my blood, you would have seen that I had polycythemia. I had higher red blood cell count than you would otherwise expect. Okay. Another condition that might cause hypoxia is hemorrhage, right? So losing blood. Also, donating blood, right? So you lose a liter of blood at a time. And so obviously for, the next hour, the next couple days, it's going to be a little bit difficult to exert yourself because you don't have enough red blood cells to transport the blood to your tissues. Okay. Also, anemia, right? So hypoxia can be a side effect of anemia, right? So anemia, is when we have less than the expected amount of red blood cells. And so we can see here that there is a much higher plasma content than there is red blood cell content. Okay. Also, natural decay. So remember that red blood cells do not have a nucleus. Remember, we kicked that out in order to create reticulocytes that can actually circulate within the blood. We need to keep our red blood cells super tiny so they can fit through our capillaries. But, the other side of this coin, the compromise here, is that without a nucleus, red blood cells cannot repair themselves when they inevitably acquire damage from the circulation process. And so these cells do live longer than the platelets, which only live a week to a week and a half. They only live about 4 months. So all the time, your liver, your spleen is filtering out these damaged red blood cells they are breaking them down, and we'll talk about that in a few minutes, and ultimately, they are going to recycle a lot of those components, right? But when you remove red blood cells from the blood, of course, you don't have as many, and so you can't transport as much oxygen. And so if you're making red blood cells and breaking down red blood cells is not balanced, this can also lead to hypoxia. Okay, so here are, essentially these vials right here, but looking a little bit more realistic. Before I move on, I just want to make sure that we understand this. I understand we're not so chatty this morning, but if anyone wants to tell me which of these is polycythemia, right? The first one, second one, or third one, I would be all ears. Right, so the question is, which is polycythemia?
The first one.
Perfect, thank you so much. Great, thanks guys. Hooray. Thanks for speaking up, I know it's super weird. So polycythemia is indeed the first vial right here. So more red blood cells than you would expect. And of course, this over here is anemia, and this is quite anemic. All right, so polycythemia, normal, and anemia. Good. All right, one more. A patient with hemophilia would benefit most from a transfusion of what? All right, so you can actually give whole blood, plasma, serum, or platelets to patients. All right, so what exactly is it that hemophiliacs need? All right, do they need, oh, Savannah, do you have an answer?
B, plasma.
Awesome, very good. So plasma, and the reason for that is that plasma contains all of those clotting proteins, okay? Serum contains no clotting proteins, plasma contains all of them. And so if a hemophiliac requires a transfusion, plasma with those clotting proteins would be ideal. Does everyone understand that? If not, you're welcome to shout out or reach out to me in office hours or in the chat that I'll check at the end. Okay. The red blood cell life cycle. Again, the red blood cells are anucleate, so they only live for about 4 months. So let's talk about what happens at the end of that 4 months. Okay, keep in mind that our circulation, our heart pumps the entire volume of blood every single minute. And so ultimately your blood is going to all different tissues within the course of an hour. And so that includes the spleen, liver, and bone marrow. And this diagram down here shows how the blood flows through the bones, flows through the liver, through the kidneys as well, and here the spleen. And so while the blood is flowing through these different organs, some of them have this filtering capability. At this point, let's focus on the spleen, liver, and bone marrow. In these structures, we have cells called macrophages, macro for big, phage or phage for eater. So these are big eaters. Their function, as actually white blood cells, is to engulf things that aren't looking quite right. And of course, this can be a bacteria, a virus, or it can be red blood cells that are a little bit worn down. Okay, so again, in the spleen, liver, and bone marrow, we have macrophages that engulf and remove from the blood worn out red blood cells. Okay, once the macrophage has the red blood cells, what can happen is that the cells start to break down within it. Ultimately, hemoglobin is broken apart. Now, it is pretty expensive to make hemoglobin, and so we try to recycle as much as physically possible. All right, first of all, we can recycle the globin. All right, remember the globin is the protein component of hemoglobin. Proteins consist of chains of amino acids, and so we can break the amino acids apart and recycle them for, more hemoglobin, or we can recycle them for other types of proteins in the body. Okay, so essentially those amino acids are dumped back into the blood, and they can be used by the body in any way it needs. Okay, a little bit more complicated than that is the recycling of the heme. Remember, the heme is the iron component. It's what actually binds to oxygen. There actually is another part of this molecule, and in the spleen, liver, and bone marrow, we can actually take the heme and break it apart. Now, the iron, which of course is acquired through our diet, right, and it's not super abundant in our diet, we really need to reuse that as much as possible. And so we essentially tack iron onto our friend transferrin, which transfers the iron back to the bone marrow, right, where it can ultimately be used in new hemoglobin. Okay, so we're not just taking, the hemoglobin and putting it into new cells, right? None of that. We're not emptying cells and refilling them with new hemoglobin. We are literally breaking down the cell, we're breaking down the hemoglobin and recycling those components to build up new red blood cells. Okay, so transferrin takes the iron back to the bone marrow to make new hemoglobin, and the one piece of this entire process that is actually just kind of eliminated from the body, thrown out, is the heme without its iron, right? And that is the pigment bilirubin. You might have heard of that before. Bilirubin, right, as we can see here, is taken from the hemoglobin, put into the blood, and ultimately it is filtered out of the blood by the liver. Okay, and so the liver is going to secrete this bilirubin into something called bile, which we'll talk a lot more about later this semester, but essentially bile is required to essentially help you absorb lipids from your diet, right? So if you eat a hamburger, all of that fat in that meat can only be absorbed into your body if we have bile. And so bile contains bilirubin, and so that's what gives bile its yellowish green pigment. Okay, so some of the bile actually goes out in the feces because of this process. Okay. Bile itself and the bilirubin component has this yellowish pigment, and so this is actually what makes the feces, for a large part, brown, right? So the yellowish tint to feces is due to bilirubin. Also, this compound, this pigment, through a process we'll talk about later this semester, can also be eliminated from the blood by the kidney, right? And so again, bilirubin has this yellowish tint to it, and so when this bilirubin derivative is released by the kidney in the urine, it also gives the urine that yellowish tint as well. Okay, so bilirubin is a pigment. It is derived from the heme component of hemoglobin. It is released by the liver and the kidney into the feces and the urine. And again, it also has a role in lipid absorption. Okay, all right, so, well, first of all, any questions about that? I know that's a lot to take in. All right, so one more question before we move along. And hopefully by the end of the week, I'll have some kind of polling system so that I can get the class's feedback about these answers as well. Okay. All right, so which of the following is false about erythrocytes? Without a nucleus, these cells have a short lifespan of about 4 months, right? That is true, right? They're anucleate, they can't repair themselves. Increasing the reticulocyte count, so the immature red blood cell count in the blood, is a normal response to anemia. This is also true, right? So if you are anemic, you have hypoxia, you need to make more red blood cells. Okay, so that is also true. Older red blood cells migrate to the liver and the spleen, where they are stripped of hemoglobin and returned to the bone marrow to be refilled. This is false, right? So this would be the answer to this question. It is false about erythrocytes. Essentially, this is false because older red blood cells aren't stripped of their contents and refilled. They are completely broken down and brand new cells are made in the bone marrow. Okay, D is indeed correct as well. Bilirubin has this yellowish tint, and so it helps to make feces brown and urine yellow. Any questions about that? All right, very good then. All right, so at the beginning of class today, I told you about Jennifer. At this point, we know enough to be able to kind of start putting the pieces together to diagnose her. This here is a graphic that I borrowed from an article published a few years ago about the diagnostic process, and we're not going to go into this in detail, and if you want, I can give you the article. It's very thorough, very good, but essentially what this is showing you is that the very first step to diagnosing a patient is gathering information, right? And so that often includes a clinical history and interview, so just talking with the patient, right? Without talking with the patient, you would never know that she's craving celery, right? You would never know what she's actually experiencing. There's a physical exam component, of course, lots of diagnostic testing, although study after study shows that really the most valuable component of this information gathering step really is just talking with the patient. Right. And finally, if you personally as a physician can't get enough information, you can refer them to another specialist. Okay, so once you have all of this information, which I've given you, and we'll talk about again in just a second, what you can then do is start to integrate that information, right? Kind of pull from your memory what you know about red blood cells, what you know about erythropoiesis, what you know in general about human physiology, and start applying these different symptoms. Is it this? Is it this? Is it this? And so ultimately, after many different kind of models that you work out in your mind, you develop what is called a working diagnosis. And so this isn't the end all be all. This is absolutely what you have 100% certain. Instead, a working diagnosis is more like the best hypothesis at the time. And so you can work on a treatment plan based on this working diagnosis, but note that the diagnostic process doesn't stop there. You will still have to gather information. You will still have to reconsider this information and reinterpret it constantly. Okay, so let's talk about Jennifer. Remember that she's losing a lot of blood. Since her pregnancy, she's really tired. She craves cold celery. Her blood looks like this as opposed to a reference blood. And here are some test results. See, first and foremost, does anybody see anything that doesn't quite look right, either from the blood test results or from the blood smear? Any thoughts at all? It's okay if not, no big deal. Again, I'm working on a polling system so that I can get some more anonymous feedback here. Okay. And of course, today I gave you the answers. That won't always be the case though. Okay, so that was kind of step one, getting the information. Step two, integrating and interpreting this information. Let's take a look at the blood test results. I give you the normal ranges for a female. You guys should become familiar with these normal ranges. On an exam, I can give you some blood test results and ask you, what's wrong, like what is off about this patient, and so you should definitely be familiar with those. Okay. All right, so first of all, the hematocrit is super low, right? So she has not quite enough red blood cells as supported by this here. So red blood cell count and hematocrit percentage in whole blood is both a little low. Her hemoglobin is also low, so she has fewer of these oxygen-carrying molecules than we would be expected to have. White blood cells, she's good, right? So this indicates that she's not fighting an infection, okay? Cholesterol, a little high, right? So maybe that's playing a role here, maybe it's not, we'll see. Glucose is good, right? Iron. Right? Iron is also a little low here, okay? And similarly, ferritin, right, the storage molecule of iron, isn't outside of the range, but it is most definitely at the bottom of said range, Okay? And vitamin B12 seems to be good, right? Remember, vitamin B12 is important in actually making new red blood cells. Okay. All right, so what's up here? Erythrocytes, hemoglobin, and iron are low, right? So whatever she's experiencing is most likely tied back to this, right? So some kind of hypoxia linked to insufficient hemoglobin, iron, and red blood cells. Okay, remember that she has, she's been losing a lot of blood due to menstruation. And of course, she's really weak since her last pregnancy. Okay. Specifically shortness of breath with exertion. When we look at her blood smear, the shape of her red blood cells, some of them are a little bit off, but not, you know, anything too crazy. But what we can see is that they are a little small, so microcytic, smaller than these nice plump erythrocytes over here. They are hypochromic. That is hypo, less than chrome color. They have less color than you would expect in normal erythrocytes, right? So these are nice and pale all the way throughout, as opposed to just a tiny little light spot in the middle from that biconcave shape. Okay, so what this might indicate is that these cells, these anucleate cells, aren't filled with the pigment, or the same amount of pigment as a healthy red blood cell would be. And so the pigment, the color, is coming from hemoglobin. Right? Hemoglobin, of course, turns red when it's bound to oxygen. And so even this blood smear is indicating that you probably have a hemoglobin problem here. Okay, so if we look back at this graphic of erythropoiesis, let me remind you that vitamin B12 is required to make new red blood cells. Remember, her B12 levels were in the expected range, so she can make red blood cells, right? She has red blood cells, and that is probably not the problem here. Hemoglobin, on the other hand, is created in these steps right here. Yes, the red blood cells exist, but we need appropriate iron in order to make hemoglobin. Okay, so if she has insufficient iron, she's going to have insufficient hemoglobin synthesis, and therefore the red blood cells are there, but they're not quite as plump and full of hemoglobin. So they're going to have a paler color without that pigment, and also they might be a little bit smaller. Okay, so as you can see, we can kind of add all of these clues, all of these different layers to our understanding of what is going on with Jennifer. Okay, and so I gave you the answers this time, right? That won't always be the case, but the working diagnosis, right, based on what we know, is that there's probably an issue with iron, and because of the lack of sufficient iron, Jennifer is having problems making hemoglobin to fill her red blood cells. And so this is actually a condition that many of us are familiar with because it's just so common in our society: iron deficiency anemia. Okay, anemia in general isn't a disease itself, but usually it's a side effect or a complication of something else that's going on in your body. In general, a lack of sufficient iron leads to decreased hemoglobin synthesis, and consequently anemia. Okay, so why might you have insufficient iron, right? What might be going on with Jennifer here? Oftentimes, this is simply reflecting an insufficient consumption of iron in your diet, right? So if you're not taking supplements, if you're not eating red meat or lots of spinach, if you don't have this nice, diverse, complex diet, you are not going to get enough iron for your red blood cells. Also, if you have any kind of stomach issues, or inflammatory issues in your digestive tract, this can very much decrease your ability to absorb iron from your diet. And so this can be linked to something like Crohn's disease. It can be linked to any kind of stomach issues. Also, if you take a lot of antacids, this can also decrease your absorption. And again, this is all tied back to the fact that in order to absorb B12 into your blood, you need something called intrinsic factor, which is produced by your stomach. Okay. Also, IDA is automatically linked to pregnant women and postpartum women, right? So people who have just given birth, by far, this is the largest population with IDA. About half of women that have recently given birth are iron deficient. And so the reason for that is that growing people, right? So a fetus or a newborn, if you're breastfeeding, requires tons of iron because they're making so much more red blood, right? And so the idea here is that, yes, they need a huge blood supply to circulate to their tissues, and of course they're growing. So, you know, in month 1, they have this volume of blood, and then 2 months later, they have 2 or 3 times that volume of blood. And so they need so much iron in order to be able to grow in a healthy way. And so as a pregnant woman, a very good portion of your iron is going to be given to the fetus. And of course, if you are breastfeeding, then a lot of your iron is given to the breastfeeding child as well. Okay, so Jennifer, of course, has recently given birth, and so this is very likely the case for her IDA. Okay. All right, so infants through 2 years old have the highest demand for iron. Again, this is all linked to the fact that baby is growing so fast. Okay. Also, females in general are much more likely to develop IDA due to menstruation. So every month, every menstrual period generally loses about 50 to 250 milliliters of blood. Of course, this is just on average. This isn't taking into account of any other bleeding issues that she might have, right? So she is automatically prone to lose a lot more iron and not be able to recycle that iron, of course, because of this menstrual loss. Okay. Also, NSAIDs, so these medicines like ibuprofen can cause gastrointestinal bleeding. And so this is bleeding that just occurs into your body cavities and isn't necessarily detected. It isn't necessarily reflected in any other symptoms, but it is happening if you're using these things for extended periods of time. And so this is one of the many reasons why you know, you shouldn't be on these medicines for many years, and why they're linked to things like anemia. Okay. IDA is also associated with fatigue and irritability. Of course, if you're not having enough hemoglobin, you don't have enough oxygen delivery to your tissues, and so your tissues are always kind of starving for that extra oxygen. And so this is associated with a lot of the things that Jennifer is experiencing, on top of sleepless nights and etc. Interestingly, the IDA is also linked to craving ice or other non-nutritive substances, such as celery, right? So if you are craving, you know, just munching on ice, or, you know, celery is nice and crunchy, but you're not actually getting a lot of calories out of it, maybe you have an iron deficiency. Okay. Okay, so most likely this is what Jennifer has. A physician would of course use IDA as their working diagnosis and treat her accordingly. Any questions about that? Again, IDA is super common in our society, so many of you are probably already familiar with it. Okay, so let's talk about some other forms of anemia. In general, anemia is just a reduced oxygen carrying capacity. Of course, this leads to hypoxia. Okay. IDA is one type of anemia. Vitamin B12 is another type of anemia. Right? Remember that we need B12 in order to make red blood cells. So if you don't have enough B12, for example, if you are vegetarian or vegan and are not taking supplemental B12, you might not be able to make red blood cells appropriately. something that might lead to B12 anemia is pernicious anemia. And so this is an autoimmune disorder usually seen in more elderly patients. And essentially their immune systems attack the part of the stomach which is responsible for making intrinsic factors. So even if these individuals are taking in a lot of B12, either through their diet or as a supplemental vitamin, they can't actually absorb it into their blood. Aplastic anemia is the destruction or inhibition of the bone marrow. Oftentimes this is a side effect of something like chemotherapy, which of course attacks any cells that are replicating really quickly, like you would see in a tumor. But of course, in your bone marrow, those hemocytoblasts are also replicating really quickly. They have to continually replace the red blood cells that are lost. And so chemo can result in anemia because essentially mitosis is inhibited throughout the body, not just in the tumor. Okay. And finally, sickle cell anemia, which is actually what one of the articles that half of you are going to read over the weekend is about. And so sickle cell anemia is a condition with a single nucleotide polymorphism. So just like you guys saw with hemophilia in Friday's lecture. This is the mutation of one single nucleotide within the DNA, and ultimately, as we can see over here, changing changing an A to a T is ultimately going to produce a different protein, so a different amino acid sequence. What this does is it makes this different shape of hemoglobin actually makes these individuals' red blood cells go from this nice biconcave disc to kind of like a horseshoe shape, so a sickle shape, when they're in low oxygen conditions. And so normal life for sickle cell anemic patients is okay unless they physically exert themselves, right? So if they're in sports or something, or if they're like, walking up a hill to get home or whatever, that is when the cells start to sickle. And that's a problem because your capillaries are essentially about as wide as a single red blood cell. And so normal old red blood cells can slip through capillaries, no big deal. But sickle cells are of course a little bit longer because of their shape, because the same cell is kind of squished. And these actually get trapped in the capillaries. And so of course blood flow stops along these capillaries, and this can be fatal in many circumstances. And so again, this is one single nucleotide mutated that leads to this very serious issue. And again, we'll talk about that a week from today. All right. Any questions about any of that? All right. We're going to start talking about blood vessel anatomy. I am not completely certain of how far I'm going to get. Whatever I don't get through today, I will record the rest and I'll post it for you to watch on Wednesday. Okay. Okay, so the blood, of course, has lots of different components, the formed elements, different proteins, other types of solutes, lots and lots of water. In general, the blood is supposed to stay inside the blood vessels. Remember that in the cardiovascular system, we have different types of organs, the heart, the blood technically is type of tissue, and the blood vessels. And as we'll see, the blood vessels vary, right? There's a couple different types of blood vessels, all of which share different layers, right? Different components, structural components, regardless of where it is in the body. I open this discussion with two what are called angiograms. So angio meaning like blood vessel, gram, so a picture, right, of these blood vessels. And so we can, of course, study blood vessels in this way. Essentially an injection of fluorescent dye is administered to the vessels of a patient, and then we can take these pretty cool images and see exactly where the blood vessels are based on where the blood is flowing. And so this is in your abdominal cavity. Here are your kidneys. Here is your aorta feeding all of your intestines, and these down here are your legs. These vessels up here are essentially, right here in your neck, and they split to feed your brain and your skull. Right, so we'll learn all about the different specific blood vessels in the lab, but keep in mind that lab and lecture in this class are very much tied together, and you should study content learned in both lecture and lab for both types of assessments. Okay, so again, blood is contained within blood vessels. Unlike some other animals, our circulatory system is closed, which essentially means that the blood is just going around in circles. It never leaves your vessels, no matter what, of course, unless you damage your vessels, and then we fix it pretty quickly. Other animals' blood actually dumps out of the vessels and gets sucked back in. Not for us. Blood stays in the vessels, and that's it. Okay. Ultimately, our circulatory system is broken into two halves, or two main circulation circuits. First, we have the pulmonary circulation, right? And pulmonary indicates lungs. And so what we can see here is that the heart pumps blue blood, and the blue indicates deoxygenated blood, pumps deoxygenated blood to the lungs. The lungs oxygenate the blood, that is, oxygen goes from the air onto our red blood cells. We'll talk about that later. And then the blood oxygenated, so now it is red, returns to the heart. And so this entire circuit right here is the pulmonary circulation. Okay, that oxygenated blood, so that red blood, is then pumped back out of the heart to the rest of the body. And I know this image might look a little weird, but essentially it is showing us that red oxygenated blood is going to the upper body, the arms, the head, etc. It's going to the liver, it's going to your digestive system, the kidneys, and your legs. Right? So all of these like mesh things here, those are capillary beds, and this diagram is just showing you that blood and its components are delivered to these different organs and then drained back to the heart. And so we can see that the capillaries are purple, indicating they are dumping oxygen into the tissues, and then the blood returns to the heart in its deoxygenated state. And so we'll talk about that more when we get into the respiratory system, but there's still oxygen in the blood, but we show it as blue just so that we can see the difference between less oxygen and more oxygen. Okay, so again, blood is always within a vessel, or the heart. It travels between the pulmonary circulation and the systemic circulation, so the systems of the body, right? The digestive system, the reproductive system, the urinary system. Okay. Ultimately, the heart itself is divided into two different pumps, right? We'll talk about this on Wednesday in your pre-recorded lecture, but there are 4 chambers, which essentially are 2 different pumps. The right side pumps to the pulmonary circuit, the left side pumps to the systemic circuit. And so note, I said that this over here, the blue pump, is the right side. Keep in mind that when we say right and left, we're always referring to patients right and left. So yes, we are looking straight at this, and my right would be the red and my left would be the blue, but the patient who, hypothetically I'm looking at, his right would be the blue and his right would be the red. Okay, so keep in mind it's always whatever the patient's right and left are. Okay. Questions? All right. Okay, so blood is always in the vessels. It spends most of its time, right? So most of the volume of the blood at any given point is within the systemic vessels. So it's within our organs, it's within our limbs, it's in our brain. And so we can see this here. This is the distribution of the blood. About 70% of the blood at any given point is in our systems, right? As opposed to the pulmonary circuit, right? Only about 18% and about 12% is hanging out in our heart, okay? Vessels themselves, right, can be named according to a couple different things. First of all, artery versus vein, right? So generally, we would think that arteries have oxygenated blood, veins have deoxygenated blood. That's not technically the distinction that is most accurate. Instead, arteries take blood away from the heart, right? Oftentimes, this is indeed oxygenated blood that's going out to the rest of the tissues. Okay, so the heart pumps blood into arteries and out to the rest of the body. Veins, on the other hand, are allowing blood to drain back to the heart. Okay, so usually this is deoxygenated blood. So the blue vessels are almost always veins. The red vessels are almost always arteries, but there are some exceptions, and we'll take a look at that later. Okay. In between the arteries and veins, we have the capillaries, and the capillaries are the thinnest vessels. They're where exchange with the tissues is actually occurring. So all the oxygen gets unloaded from the capillaries to the tissues. All the sugar, all the vitamins, all of these things get unloaded from the capillaries, not from the arteries or the veins. Okay. All right. The other factor that we use to classify blood vessels is the size and the structure of the vessels. And so that's what the next couple slides are going to be here. Before we move into them, I do want to point out that the veins are sometimes called capacitance vessels because at any given moment, about 50% of your blood is in your veins. And so we'll see that the veins are really low pressure conduits. And so it takes a long time for blood to go from your feet to actually like get milked all the way back up to your heart. And so you'd think that that would be detrimental, but in fact, you don't need all of your blood circulating when you're just kind of, chilling here watching a Zoom lecture, right? But if you get up and you go for a jog right away, all of that blood that was kind of hanging out in your veins can now be mobilized really quickly. And now you have increased your oxygen carrying capacity so that you can support and sustain that extra activity level. Okay, so these are capacitance vessels. They have a lot more blood in them at any given point, but that can be mobilized really quickly the moment that you need it. Okay, arteries and veins. Right, arteries going from the heart, veins going to the heart. All of these vessels have the same 3 types of layers within their vessel walls. And this is something that you guys should get used to. Pretty much every organ that we're going to study this semester has 3 layers making up the walls, right? So with a couple exceptions, but for the most part, 3 layers make up the walls. The first layer, all the way on the outside, so the most superficial layer of these vessels, is connective tissue, right? This is essentially like securing the vessels to the surrounding muscle, the surrounding fascia, keeping them in place so they don't like wiggle all over the place. Okay, we can call this the tunica externa. Tunic means like layer, right? So yeah, literally a layer. Externa, this is the most external of all of the layers. Sometimes this layer is called the adventitia, depending on where we're talking about. So for example, the adventitia covers our esophagus, but not a lot of other organs. Okay, so I will make that distinction as we go forth. In general, the outermost layer of the blood vessels is the tunica externa. Next, we have the smooth muscle layer. This is the tunica media. Okay, as we can see, this is quite variable between veins and arteries. We'll talk about that in a moment. But this is the middle layer. And as we can see, the smooth muscle cells kind of go around and around this vessel so that we can actually change the diameter of these vessels. And that's going to be really important within the next couple days. Endothelium, right? So this is the special word for the epithelial lining of the blood vessels. This is simple squamous or squamous epithelial tissue. It allows a lot of passage of substances through it. Right? It's a very porous, but not for certain things. We'll talk about that too. The endothelium, the layer itself, is called either the tunica interna, right, the most internal layer, or the tunica intima. Okay, depending on which textbook you're looking at, you will see both interna and intima. Either one is okay for me. Okay, so again, the interna is lining the blood vessel that's made of endothelium. Smooth muscle is next. That is the tunica media. And finally, the tunica externa is all the way on the outside. Okay, keep in mind that the third type of vessel is a capillary. And because capillaries are where all of this exchange happens with the tissues, they don't have the tunica media or the tunica externa. Instead, they only have the endothelium or the tunica interna. All right, so let's talk a little bit more about these layers. The outermost connective tissue layer, the tunica externa, is largely loose connective tissue, again, just kind of securing these vessels to their surrounding tissues, protecting them as well. In certain vessels, the tunica media, so the smooth muscle layer, is so thick that it has its own blood supply and its own nerves. Okay, so what we can see in this image here is the tunica externa, and this squiggly stuff right here is the smooth muscle. And what we can also see is that we have a blood vessel in red and a yellow nerve that are actually innervating and feeding the tunica media. All right, so this is only the largest of all the blood vessels, but yes, the blood vessels have their own blood vessels. And so we call that vasa vasorum, which in Latin is literally vessels of the vessels. Okay. Next, the middle layer, the tunica media. This, of course, is the thickest layer. It consists of smooth muscle. Essentially, this smooth muscle, depending on how thick it is, can keep open vessels, right, as opposed to like ones that collapse when there's not a lot of blood in them. So it can keep the vessel open. The smooth muscle itself can provide vasomotion. So essentially, not only does your heart beat to move your blood along, but also your vessels have their own intrinsic beating rhythm. And so this helps to make sure that all of your blood is constantly in motion. And of course, when you get up from our Zoom call and get out and take a jog, we need to mobilize that blood really quickly. And so one of the ways we do that is through this vasomotion, right? So lots of contraction all at the same time, so blood keeps circulating quickly. This vasomotion happens at a particular rate when you're younger, and over the course of your life, it gets slower and slower and slower. And so that's one of the reasons why elderly patients start to have more circulatory issues because they have less vasomotion. Okay. Also, we can control our blood pressure by changing the diameter of our vessels, right? So in general, large vessels let blood flow freely, and so there's less pressure. If we constrict those blood vessels, there's a lot more blood pressure. And so we're going to spend a lot of time over the next couple weeks talking about vasoconstriction, making them smaller, and vasodilation, making the diameter bigger. And so all of this is this change in diameter is created by controlling the tunica media. Okay. Finally, the innermost layer, again, simple squamous epithelial tissue, very, very skinny cells. This is what is physically interacting with the blood. And so in the last lesson that you guys had, we talked a little bit about when the endothelium is damaged and how hemostasis occurs and the platelets are essentially binding to those exposed collagen fibers. All of that is happening within the tunica interna, right? Let's see, also this is a selectively permeable barrier. It generally only allows really tiny things like sugar and gases and vitamins to actually cross over. It never allows red blood cells to cross over unless it's damaged. but healthy tunica interna keeps the blood inside. All right. Arteries as opposed to veins. In general, arteries are more muscular than veins are. Okay, so when we look at these two histological slides, we can see that the tunica media, so this squiggly pink with purple dots section here, this is a really thick tunica media as opposed to a much skinnier tunica media up here in the veins. Also, here a relatively thick tunica media and very skinny over here in the vein. Arteries are able to resist a higher blood pressure than the veins are. And so this is very much tied back to the fact that they have a thicker tunica media. They also have a lot stronger connective tissue around the outsides. And so veins are called capacitance vessels because they have the capacity to hold a lot of extra blood at very low pressure. The arteries, on the other hand, are sometimes called resistance vessels because they can resist a huge change in blood pressure. And again, something we'll talk about quite a lot. The arteries, as opposed to the veins, maintain their round shape even if there's no blood inside them. So as you can see here, there's no blood in the lumen or the opening in the artery or the vein. The artery maintains this lovely round shape, right, very strong. The veins, on the other hand, kind of collapse when there's not a lot of blood in them. Okay, so it's relatively easy to tell the difference between the two. Even in histological slides, you will be looking at those in the lab. All right, finally, arteries are divided into 3 categories according to their size. Okay, the first one that we can talk about are the conducting or the elastic arteries. These have by far the largest diameter. Right, and so examples of these are the ascending aorta, right? So the blood vessel that's coming right out of the left side of your heart withstands huge variance in blood pressure, so it has to be super strong and always remain open. It has a very thick tunica media, and it has a lot of elastin and collagen inside, so it's really reinforced so that it can stretch and recoil with every single heartbeat. Okay, so it's distensible. It is a pressure reservoir. So if your aorta or any other of these conducting or elastic arteries develops plaques, so atherosclerosis plaques, essentially those vessels are no longer able to stretch and recoil to the same extent, and so they just remain the same diameter. And so this is a problem because every time your heart beats, these vessels are taking in huge amounts of pressure, and they can't even stretch to like reduce the pressure on themselves. They're just getting the brunt of the pressure generated by the left ventricle, and they just have to withstand that. And so for the next lesson, I will finish up talking about this particular content, so to be continued, and we will pick up with talking about atherosclerosis and how it really damages your arteries and how that can lead to aneurysms. Right.